meaning of these traits in terms of how they are transferred from, say, the mother to the offspring. And this is just a picture showing a cell undergoing cell division, right? You can see the chromosomes are staying nicely in blue color, and you can see how the chromosomes are being pulled towards the opposite sides of the cell so that they can divide in the middle and you get two daughter cells. So this is all people could observe, scientists could observe in the 1800s and even in the early 1900s. All they saw was chromosome. But however, the big question is, what is this chromosome made up of? So today we're going to look at what is chromosome made up of, and I'm sure you all know what are chromosome made up of. DNA, right? And so if you want to look at a cartoon representation, people know that in non-dividing cells, the chromosomes are in the form of this spaghetti. They're kind of in an extended state. They're not condensed like doing cell division, but they're condensed to form visible chromosomes or chromatids. Okay? Whereas in non-dividing cells, generally, the chromosomes are all spread out in this sort of double-stranded structure. And we're going to look at the structure of DNA later on. Okay, so the question is, what is this structure? And so one of the biggest discoveries in the 19th century was discovering or solving the structure of the DNA. You know the name of the scientists, Watson and Crick? Have you heard their names? Yeah? Actually, Crick used to live here in La Jolla. He passed away a few years ago. He was working at the Salk Institute. And Watson is still alive. And they, in 1953, they proposed what the structure of this chromosome is, or what the structure of DNA is. And this is their very famous model. They're showing the DNA. DNA is made up of how many strands? Can somebody count? Two strands, right? You must have learned this in your school. And so, if you sort of look at the cartoon's representation, if you take the chromosome, if you unwind it and look at just a single molecule of DNA, it's actually made up of two strands, and there are bases, which we will describe later on, that are on the inside of these strands. And those bases are the ones that code or represent the various traits, like for example, pink flower versus white flower, things like that. All that information is packed in DNA. So for proposing the structure, the double helical structure of DNA, Watson and Crick, along with Maurice Wilkins, got the Nobel Prize in 1962. So why is DNA structure so important? And feel free to stop and ask questions, okay? I tend to go fast. Okay, so you might already know, and you also mentioned, DNA is made up of two strands. So it forms a helical structure, so that's why it's called a double helix. And the other important thing for you to remember for your science Olympiad is that the two strands are anti-parallel. What does that mean? Anybody knows what anti-parallel means? Yeah?
They aren't parallel.
They aren't parallel. But they are, in some ways, are parallel to each other. But what is meant by anti-parallel?
They like intersecting each other?
No, they don't intersect each other. So you can see how they're like this. There is, it might be a little bit more advanced. Okay, tell me.
It's on a regular two-axis Cartesian plane, it won't be parallel, but on a three-dimensional Cartesian plane, as you...
No, it's nothing complicated. It's more it's more biological, this definition. So I will come to that in a minute. But basically, when you have two strands and there is directionality, one is going what we call 5' to 3', okay? Because of the asymmetry in which how the molecule is formed, you have one going 5' to 3', and the complementary strand will be going in 3' to 5', so in opposite direction. And that's why they call it anti-parallel. I'll show you a cartoon of what I mean by anti-parallel. And the other important thing is that DNA is made up of nucleotides, right? And the nucleotides in turn are made up of three building blocks. This is sort of an overview slide that I stole from the internet from somewhere. I think it was for Science Olympiad. And I have better slides organizing what the building blocks are. But basically you have sugar, phosphate, and base. So if I were to write it down, I want you to write these things down. Write down, there are three components for DNA. One is base, B-A-S-E, base, okay? The second is phosphate. There's a phosphate group in DNA. And the third thing is there's a sugar molecule. So those are the three things that come together to form DNA. Clear so far? And I will explain what this base, sugar, and phosphate groups are. And you need to know that much. Now the bases in DNA are given the names listed down there: adenine, thymine, guanine, and cytosine. Okay, or adenine, thymine, guanine, cytosine. So write those things down. You don't have to write actually. If you want, you write, because I'm going to post all this. Eventually when I figure out, I'm going to post it online, or I'll email you the slides. Okay? But these are the 4 bases in DNA, and they are the sort of the code for how the DNA is able to store information. For example, in computers, you have zeros and ones. And with zeros and ones, you can store all the information in the computer. In the same way, for DNA, there is a 4-letter code. So we abbreviate it as A, T, G, and C. Okay. So, to refresh your memory again, the simplest building block of DNA and RNA is called a nucleotide. Okay? And the nucleotide is made up of 3 components. There's a base, and it shows here a base. There's a sugar, and then there is a phosphate group. I'm sure you don't want to learn the structures, okay? This is chemistry. You don't have to memorize the structure. All you have to know is that there's a base, there's a sugar, there's a phosphate group. They're all attached together in this form. So you have this C1 prime carbon attached to a nitrogen base. In this case, we're looking at a heterocyclic base, which is actually a cytosine, and is attached to a phosphate group at carbon number 5. Okay, so this is a phosphate group that's sort of color-coded. Just to reinforce the concept of what they're made up of. Okay. What is the difference? Have you heard the word RNA? Just like DNA, there's something called RNA. What is RNA? What is it?
Is it the instructions for making proteins?
Yeah, so RNA is the one that's actually contains instruction for making protein. DNA is like the central library. It's got all the information. It's in the nucleus. But you don't carry the whole book and take it home with you, right? You only take 1 or 2 books. And those 2 books or more is the RNA. That is where small snippets of the DNA sequence is transcribed, okay, or copied to make an RNA. And then the RNA is finally used to make proteins by these machines called ribosomes, okay? So now we're going to look at the difference between DNA and RNA. DNA and RNA are very similar, okay? It's hard to tell them apart. There's some very small differences between them. One is if you look at the sugar. If you look at the sugar, in the case of RNA, we call it ribose. That's why it's called ribonucleic acid. What is DNA? What is the expansion of DNA?
No oxygen in the ribose.
Yeah, but what is the expansion? We call DNA, D is standing for deoxyribonucleic acid. That's DNA. Everybody says DNA. Even a layman knows what, use the word DNA. But the actual reason we call it DNA is because of the deoxy meaning missing an oxygen. Okay? So if you look at this carbon number 2, it's missing this oxygen. It only has 1 hydrogen here, no oxygen. And that gives DNA this extraordinary stability. So you might have seen the movie Jurassic Park. How many of you have seen Jurassic Park? And there they got DNA from what?
The fossil.
Amber, right? Some amber. Basically they got it from tree gum, which forms amber. Inside that there were like mosquitoes which had sucked blood from some dinosaurs, so they were able to get the DNA. So that's how the story goes. So DNA, that tells you that DNA is very stable. It can be there for 65 million years, and they can still purify the DNA and read the code. On the other hand, RNA, because of this 2' hydroxyl group, is very reactive. It's not very stable. And the cell doesn't want it to be stable because it makes its short messages makes the protein and then gets rid of the RNA. Whereas DNA has all the information stored because it's a genetic material that needs to be transmitted from one generation to the other. And so that's why it's missing this oxygen, which makes it more stable. Okay, so important thing to remember, RNA has a hydroxyl group at carbon number 2, okay? That's why it's called ribose, whereas DNA is called, the DNA sugar is called deoxy because it's missing that oxygen. Does not have the 2' hydroxyl group. Any questions so far? Okay, so now you know the sugar in RNA and DNA. We're going to look at the bases. Can someone tell me, is there any difference in the bases between DNA and RNA? Yeah?
Instead of thymine, it's
Uridine or
uracil.
uracil. Okay, uracil, very good, excellent. Okay, so that's one small difference. So normally in DNA you have adenine, guanine, that's why it says DNA and RNA, and then you have cytosine, both in DNA and RNA. But in DNA you only have thymine, okay? Whereas in RNA you have uracil. What is the difference between thymine and uracil? It's a small difference. In carbon number 5 you have a methyl group, so it's called thymine. Whereas if you don't have that CH3 methyl group, it's called uracil. Okay, that's one way the cell can distinguish between DNA and RNA is by checking the base and saying, hey, this is thymine, so it must be DNA, whereas this is uracil, so it must be an RNA. So it's down there, I've written down the various bases in DNA, adenine, guanine, cytosine, and thymine. And this one is important for you to remember for your science Olympiad, okay? In the case of RNA, remember, it's uracil. So what did we learn so far? We learned about a phosphate group that's common between DNA and RNA. There's no difference, okay? On the other hand, the sugar, there is a small difference. In the case of RNA, what do you have at the carbon number 2?
And extra oxygen.
You have an extra oxygen, we call it a hydroxyl group, okay? In the DNA, you don't have that. In case of bases, what is the difference between DNA and RNA? Thymine in DNA and uracil in RNA. So that's the only difference. You don't have to memorize the structures or any of those things, okay? This just shows you how you number the various carbon and nitrogen atoms inside purines and pyrimidines. So adenine and guanine, these big ones, are generally called purines, P-U-R-I-N-E. I don't know whether they will ask you that. Whereas the small ones, like cytosine, thymine, and uracil, they only contain one heterocyclic ring, and they're called the pyrimidine base. Okay, so purines and pyrimidines. Maybe they might ask you, maybe they may not, but I want to cover that. Okay. Question now is the chemistry. How do you attach these various groups together to form DNA? Because DNA is not just one nucleotide, it's a long chain. Some of this can be thousands, maybe 10,000 nucleotides long. So how do you form these things? So you form a nucleoside, okay, not a nucleotide, but a nucleoside when you attach a base to a sugar. In this case, we're taking a ribose sugar and we're attaching adenine, okay? And we're getting something called adenosine, okay? And the bond that's formed is between the nitrogen 9 and carbon C1 prime. We call this a beta and glycosylate bond. So this is, without the phosphate, we call it a nucleoside. With the phosphate, we call it a nucleotide. Okay? They won't ask you how to link them together. This is an example of uridine. Okay? Here the uridine, the uracil base is attached to a, always check this position. It's a hydroxyl group, so it has to be an RNA. So it's a ribose sugar. And here is an example of a deoxythymidine, where you have a thymine, see the methyl group here, carbon number 5, attached to a sugar, and it's missing the hydroxyl group, so that's why it's called deoxythymidine. This is just to reinforce what we just learned. Questions so far? Clear? Very simple, right? 3 components, you learned them. And we know the 4 bases. Okay, they're going to go one more step. You want to go from a nucleoside to a nucleotide. What is the difference? You're just going to attach a phosphate group at carbon number 5. this is very important when you start forming long chains. So when you add the phosphate group, it's a 5' monophosphate, and you call that a deoxyribonucleotide, TIDE. Okay? So going from a SIDE to a TIDE, then you attach a phosphate. Now these are all chemical structures. In real life, if you look at the molecule of ATP as shown here, this is what we call a space filling model. This is the actual volume occupied by various atoms in ATP. This is how the shape would look like. Okay? They occupy some volume. Adenosine triphosphate, excellent. What is ATP so important? It's so famous for? Energy, yeah, it's a source of energy in all living things. We all store ATP, like a person would consume almost the same weight in ATP in in a day or so. So it's a lot of energy being burned. And ATP is the energy currency of the cell. Okay, and this is just a table for you to reinforce the same concepts. You have the bases listed on the left side, DNA and RNA. When you have a nucleoside, meaning it has got the sugar and the base attached, that's the name that you give it, deoxyadenosine, deoxyguanosine. If it's RNA, it's adenosine, guanosine, things like that. And when you attach the phosphate, then you say where the phosphate is attached. You call it adenosine 5' monophosphate, phosphidine 5' monophosphate. So that's when you attach all the phosphates, you call it a nucleotide. So it's the same thing. Any questions? Not too fast? Okay. Now we learned how to form a nucleotide, and we have to link them together to form a chain. Okay, DNA is a long polymer of nucleotides. And basically you bring this phosphate, you attach 5' to 3'. Okay? You see there's a phosphodiester bond formed. And this chain can continue so on for thousands of nucleotides. Okay, so this is a dinucleotide because we have attached two nucleotides together and we call it a di, meaning two, dinucleotide. And if you attach this and form a very long chain, you can have something like this. You can have one nucleotide, another nucleotide, another nucleotide. They all linked by a phosphate group in the middle. Okay? So if you look at this cartoon, I suggest just remember this cartoon. You have phosphate, sugar, and to the sugar is attached the base. Then the next one, another phosphate, sugar, base. And they're linked via this phosphate, sugar, phosphate, sugar, phosphate, sugar. See, phosphate, sugar, so on. And so that gives directionality. Remember I asked you a question about parallel versus anti-parallel. This is the 5' carbon, right? So we give this end the 5' position. We call this is the 5' end of the DNA. And this bottom piece is the 3' carbon. So we call this the 3' position. Makes sense to you? 5' position, 3' position. So this chain is going 5' to 3'. Clear to you? And when the another strand comes together, it's going to go in the opposite direction. It's going to go from 5' to 3', going from bottom to up. And that's why we call it anti-parallel, because the directions are reversed when the two strands come together to form a DNA duplex. Okay.
Question.
Yeah.
Oh,
Go ahead.
I
Okay.
have the question, sorry. What was the long chain called?
It's a polynucleotide. In this case, there are three, so we call it a deoxytrinucleotide.
Oh.
But normally we call it a polynucleotide. Okay, poly meaning many nucleotides together is called a polynucleotide. DNA is always a polymer, so it's a polynucleotide. I don't know whether you know about protein structure, but proteins generally are made up of, what are proteins made up of? Amino acids, right? And DNA is made up of, we just learned, nucleotides. And so I just told you that nucleic acids, you always start from the 5' end and go towards the 3' end. That's how you might have seen in movies, they have all these DNA sequences and they usually read it out. And when they read it out, it goes from 5' to 3'. That's how scientists like to represent the DNA sequence, okay? On the other hand, proteins also have directionality, protein chains are always written from the amino terminus, in short form we call it N-terminus, towards the C-terminus or the carboxy terminus. This is just showing how the backbone of a protein is and how the backbone of a DNA is. You don't have to memorize any of these things. Okay, cartoons are much more useful. Let's look at why DNA is a double helix. Okay, so now on the left we have one chain of DNA, a polynucleotide, going 5' to 3' because this is where the 5' phosphate is and this is where the 3' hydroxyl group is. The bases are all on the inside. And these bases, they don't like to remain single-stranded. They don't like to be single. They want to pair always. Okay? So what do they pair with? They pair with another DNA. But for them to come together, they have to be in opposite direction. So here you see this is going 5' to 3'. 3' hydroxyl group is out here, the 5' phosphate is here. And when they come in this sort of anti-parallel fashion, they can form a duplex. Okay, that's a DNA duplex. These dots are representing hydrogen bonds between thymine and adenine. Okay, and guanine and cytosine can form a more stable base pair because they can form 3 hydrogen bonds, whereas thymine and adenine can only form 2 hydrogen bonds. So GC base pair, TA base pair. They always go together in DNA. Okay, A likes to go only with T. It won't go with G or C. And G only goes with C. They're like made for each other. Is that clear? Okay.
Don't some mutations happen when a G combines with a T or something like that?
Yeah, during replication, we're going to talk about DNA replication. Sometimes the enzymes are not perfect, they're not 100% accurate. When they're making a copy of this, instead of putting a C opposite to a G, it might put a T. And then you have a mutation. And then that can go on from generation to generation. So that is due to errors during DNA replication. Sometimes there might be chemical agents. If you smoke, if you take drugs, if you drink alcohol, some of these chemical agents are carcinogens, excessive sunlight, they can damage the DNA. Okay, if the DNA is damaged, there are enzymes in our body that will repair the damage. We are constantly damaging our DNA. There are enzymes that goes in and fixes the DNA. And when they fix the DNA, they may make a mistake, and that can also cause a mutation. Okay? So there are 2 ways you may end up with, say, cancer or something like that. One is if you get a mutation during DNA replication, that is when the child is born, things like that, when the gamete is dividing. Or if you expose yourself, that's why they tell you put sunscreen when you go out in the sun or go to the beach or something like that, because you want to protect your skin from UV radiation. UV radiation can damage our DNA, and that can result in mutations which can cause skin cancer. Okay, clear? Okay, so here we are looking at how the 2 strands come together. But this is just a cartoon. In reality, you can also represent it in this form, sort of a simplified form in this ladder structure. Okay, but DNA does not like to remain in this plain ladder-like structure. It likes to actually twist around itself to form what we call a helix, as shown here. So this is the actual structure of DNA. DNA is not in a linear ladder, and it always forms a helical structure. It's more advanced. I ask my students, I teach at UCSD, I ask my students, why does DNA like to form a helix? Nobody knows the answer. And they are college students. So if you don't know the answer, it's okay. Why do you think DNA likes to form a helical structure? Why not just a plain ladder? Okay, go ahead, you can guess. I don't.
Okay, I'm gonna take a random guess.
Yeah.
Is it because it takes up less space, or?
It takes up less space, that's a good reason. That's actually one of the principal reasons underlying it, okay? Yeah?
I actually have 2 reasons. Because the hydrogen bonds may be a little loose somewhere,
Little what?
there may be 2 hydrogen bonds somewhere, or 3 hydrogen bonds, or it could be that on one's the double helix, since they're made of 2 strands, since one is going one way and the other is going the other, that's what's causing the twisting.
Good, think It's good to think, but the actual reason is very simple. I'll tell you the reason. If you look at the nitrogen bases, you know the bases, we looked at the bases, what are they? They're big organic molecules, right? They're like big structures. Those structures, they don't like water at all. Okay, they are hydrophobic, what we call hydrophobic, meaning they hate water. And so to minimize exposure with water, when you twist them, like she said, you sort of tighten the space. When you take a rope and twist it, you reduce the space. So less surface area of the base is exposed to water. And they're trying to avoid water at any cost, and that's where they take this sort of a helical twist. Okay? They're very hydrophobic. On the other hand, you know the sugar. When you take sugar and put it when you take a teaspoon of sugar and put it in water, does it dissolve? Dissolves, right? Sugar is hydrophobic or hydrophilic? Hydrophilic. It loves water, right? Philia means love in Greek. So hydrophilic means it loves water. And so that's why nature has created this amazing molecule with sugar and phosphate. Phosphate is also charged. It loves water. So these things make the outside of the DNA hydrophilic, and they can really dissolve in water, whereas the bases are all tucked in and they're protected, okay, by being tucked in and by forming these hydrogen bonds. So you need to know that DNA is made up of two strands. They run in anti-parallel fashion. What is meant by anti-parallel? It means 5' to 3' on one strand, 3' to 5' on the other strand. You should also know how the base pairing method works. So what does adenine base pair with? Thymine. And what does cytosine base pair with? Guanine. That much you must remember. In the case of RNA, thymine is replaced by uracil, so adenine will base pair with uracil, okay? And guanine will base pair with cytosine. So here are some more pictures of DNA. You might have seen this in textbooks and various other magazines, for example. People represent it as what we call CPK. This is called where each of the atoms are color coded. So you have carbon is probably in silver color. We have oxygens in red color and nitrogen in blue color, things like that. So that is just for clarity. This is how the actual DNA by volume will occupy. This is called a space filling model. And this is, of course, a cartoon representation just to illustrate how the base pairing schemes work and how the backbone is always on the outside and the bases are always on the inside. That's all for DNA. We're going to look at DNA replication. Any questions so far? Okay, DNA replication. Wow, we've got to go fast. So now replication is, what is replication? You need to make a copy. You need to replicate, right? You need to take one double-stranded DNA and make two out of it. So your syllabus says that you need to know that replication, basically there are few terminologies that during replication, synthesis always takes place from 5' to 3' direction. The enzymes can synthesize only in 5' to 3' direction. And there is this terminology called Okazaki fragment. And we'll come upon what Okazaki fragment means in a minute or so. So if you look at the DNA that's replicating, you see we are going from one double-stranded DNA to two double-stranded DNA. Okay, so you're replicating, you're making a new blue strand here. And the place where replication is actually taking place is called a replication fork. Okay, that's called a replication fork because it looks like a fork. Okay, and the enzyme. Anybody can tell me what is the enzyme that replicates DNA?
RNA.
No, enzyme.
Oh,
Enzyme is a protein that makes DNA.
Oh,
Another Nobel Prize. This was for Arthur Kornberg. He was the one who isolated this enzyme that makes DNA. It's called DNA. It's very simple name actually. DNA polymerase. What is meant by polymerase? Polymerase is an enzyme that can polymerize or make long chains out of building blocks. Okay. So the name of the enzyme is DNA polymerase. Okay. So the idea is this: you take a double-stranded DNA and you're going to make two daughter DNA duplexes. I don't think I need to explain semi-conservative replication, that's more advanced. So let's look at the replication fork. If you look at the cell which is actively dividing its DNA, what you will notice is these kind of things. In the DNA double helix, you start seeing bulges. The DNA is opening up, so the enzymes can bind and they can start making copies. The enzyme is shown here in purple color. The enzyme is a DNA polymerase. It will bind and then use the information in one strand of DNA to copy and make another strand. So it'll end up with two daughter strands. Photos. So this is an electron microscopic image of a replicating DNA or of a replicating chromosome. I think this is a bacterial chromosome. You can see all this, right? Can you see all these big round structures, openings? Yeah, that is where replication is taking place. The DNA strands are separated, the enzymes are sitting there, and they're making a copy of a new strand. And that's illustrated here. So you have these purple enzymes. They are at each of these replication forks, and they're copying the double-stranded DNA, and they're making a new strand. But there is one problem. I don't know how to explain it to you. There's one problem, and that since the DNA strands are running in opposite direction, this strand is going, say, I guess, in a 5' to 3' direction, and the other strand is going 3' to 5' direction. Always one strand, the synthesis is easy, because synthesis always goes in 5' to 3' direction, okay? Whereas in the opposite strand, synthesis is a little bit difficult, because only small portions of the DNA is available to the enzyme for synthesizing the new strand. So we have something called leading strand and lagging strand. In leading strand, the synthesis is continuous. The enzyme can just keep making the daughter strand. Whereas in lagging strand, the synthesis is discontinuous, meaning you make small pieces, and then in the end, many enzymes will come and join all the small pieces together. What do you have to remember? You just have to remember that the small DNA fragments that are made during replication is called Okazaki fragment. What is it called?
Okazaki.
Okazaki, after the scientist. So go back here. Remember this thing, Okazaki fragment. Okazaki fragment, the small pieces of DNA that are made during DNA replication for the lagging strands. These small pieces are made, and this is the Okazaki fragment. Whereas the other strand is synthesized continuously, okay? That's due to how the DNA strand is anti-parallel. So remember Okazaki fragment. Okay. This is again showing a replication fork. It's saying how the DNA opens up. There are enzymes that are synthesizing. This is a leading strand, so the enzyme can continuously keep synthesizing as the DNA opens up. But here it has to wait. A little bit opens up, it will synthesize a small piece, another small piece, another small piece. Each of the small pieces are called Okazaki fragment, okay? And then DNA ligase will join them together. So that's all for DNA replication. We're going to quickly look at the poor cousin of DNA, RNA. So RNA was not very well, it's not as famous as DNA. Everybody talks about DNA, but now RNA is known to play very important roles. What is the difference between DNA and RNA? Now, RNA generally is single-stranded compared to DNA, which is always double-stranded, right? And we already know it's ribose in RNA and deoxy in DNA, uracil instead of thymine. Now, there are different types of RNA. You have messenger RNA. What does messenger RNA do? Anybody knows?
Carries messages.
Carries information from DNA to the cytoplasm in the cell, where that information is used to make protein. What about transfer RNAs? Have you heard about transfer RNAs?
Yeah.
What do they transfer? And why do you need amino acids? To make protein. So you guys know everything. Good. What about ribosomal RNA? Where is ribosomal RNA found?
Ribosomes.
Part of the ribosome. They carry out very important catalytic function in the ribosome. Okay. So those are the different types of RNA you need to know. You need to know mRNA, tRNA, and ribosomal RNA. Okay. Now we learned about DNA replication, and very much similar to DNA replication, there's a process called transcription. Again, you make use of the DNA information and you make a copy of RNA. What is the enzyme that does transcription? Anybody knows? Similar to DNA polymerase. You can guess. RNA polymerase, but scientists are not very imaginative. They use the same words again and again. So it's RNA polymerase. And finally, you know what translation is, right? Translation is nothing but translating the information in mRNA to make proteins. It's also known as protein synthesis. Okay.
Wait, what is translation again?
Translation is protein synthesis. That
Oh,
is
okay.
using the information in mRNA to make proteins by the ribosome is called translation. Okay. This table again tells you what the difference between DNA and RNA is. We won't go over that. So where is the DNA in the cell always? Which part? Which part of the cell? In the nucleus. Then where does transcription take place?
Nucleus.
In the nucleus, because DNA is in the nucleus. What about protein synthesis? Where does that take place?
Some.
In the cytoplasm. So you have the nucleus and then you have the cytoplasm, right? So you have ribosomes, and ribosomes are all in the cytoplasm. So you need to make a copy, you may need to make a small mRNA in the nucleus and then take it all the way into the cytoplasm so that the ribosomes can read the mRNA and make the appropriate protein. Okay. So if you look at transcription, you don't need to memorize anything. All you need to know is it's very similar to DNA replication. The two strands will separate and this enzyme called RNA polymerase will bind and synthesize from 5' to 3'. Always synthesis is from 5' to 3'. You will see synthesis of a new RNA strand, and that process is called transcription. That's another nice colorful picture here. So you have your DNA in the nucleus, the double strand, and you have a gene. So you remember Mendel, he didn't know what a gene was for tallness or for flower color, you know, pink versus red versus white. There are information in the DNA, and that information needs to be transcribed so you can make the pigments in the flower. So how do you do that? You separate the two strands, and then the strand is read by an RNA polymerase, which makes an mRNA, and then the mRNA is translated in the form of codons by the ribosome. So that's just telling you how the DNA strands separate. Okay, again, DNA, make an mRNA, the mRNA leaves the nucleus, goes to the cytoplasm, and the cytoplasm, with the help of tRNA, which has amino acids attached to them, the ribosome can synthesize the polypeptide in the cytoplasm. Anybody heard of the genetic code? What is meant by the genetic code? No? Okay, so how does the ribosome know which amino acid to add? Okay? When it's making a protein, it needs to know, it needs an information transfer, right, from the DNA to the amino acid. So that's called the genetic code. Somehow, taking the DNA code and using that to make proteins requires translation. That's where the process is called translation. So for example, we have what we call a codon, a 3-letter code for each amino acid. For example, phenylalanine is an amino acid. The codon is UUU. So like this, you can have 64 possible combinations, right? How many amino acids are there in the cell? Huh?
23.
Generally, common amino acids, how many are there? No?
Millions.
No. Somebody said 20, which was good. 20, 20. 20, okay? There are 20 amino acids. What are the 20 amino acids? You can, it's all listed out here, there are multiple copies of it. However, there are only 4 bases, right? There are only 4 bases and 20 amino acids. That's why you need a 3-letter codon. That is, the 3 letters, you can pretty much specify easily 20 amino acids, right? Yeah?
Sorry, what were the 3 RNA types that we needed to know about?
Messenger
Yeah,
RNA,
that was-
okay? Transfer RNA. And finally, ribosomal RNA.
Thank you.
Okay. So the 3-letter code for amino acid is called codon. So UUU is for phe, and for example, AUG is for med, GGG is for glycine. So things like that. You don't have to know, all you need to know what is the genetic code is basically the code that is used to represent, the 3-letter code that is used to represent an amino acid is called the genetic code. So it's a 3-letter code used to represent an amino acid. And this 3-letter code is actually read by tRNA. So this is the structure of tRNA molecule. It has this cross-like structure. The actual structure in 3D is this L-shaped structure. What is important is that amino acids are attached to the 3' end of the tRNA molecule. So this is an amino acid attached to the tRNA. You can see an amino acid attached to a tRNA. And this is what that binds to the ribosome during protein synthesis. So that is tRNA. This is a picture that I made of the ribosome. So ribosomes are these huge machines. Yeah, go for it.
The tRNA is the transfer RNA, right?
tRNA is transfer RNA. T is abbreviated for transfer. Okay? M is for messenger. R is for ribosomal. Okay. So tRNA is the substrate, or tRNA is the molecule that brings the amino acid to the ribosome, and it has something called an anticodon. Remember Watson-Crick base pairing? So this anticodon of the tRNA will form base pair with the mRNA, and that's how the information is translated. I think that's more advanced for you. Someone just remember tRNA and it brings amino acid to the ribosome. That's good enough. Okay. This is just a nice picture of the ribosome, how it really looks. This is the crystal structure of the ribosome. And this is from a textbook. I don't think you need to know this. All you need to know is that tRNA will bind to the ribosome in a stepwise fashion. So tRNA will come, bind to the ribosome one at a time, then bring an amino acid to the ribosome, and the ribosome will catalyze peptide bond formation. See, 2 tRNAs are lined up properly, they're interacting with the mRNA, and then the mRNA is going to catalyze peptide bond. So amino acid 1 is attached to amino acid number 2, as shown here. And this one will go up, a new tRNA will bind with amino acid number 3, then again the ribosome will attach it to a growing chain, then 4 will come, so on and so forth. So the ribosome is just like an assembly machine, okay, just like a car assembly machine. It's taking the tRNAs and attaching the amino acids together. Clear so far? It's a very complicated process. My lab, I have a lab, and we study this whole process. How does the ribosome do this and all that stuff, okay? This is sort of a cartoon that I made talking about various factors that help the ribosome in catalyzing protein synthesis. So again, it's the same idea. You have a tRNA bound to the ribosome. This is the ribosome. Amino acid is shown here. The new tRNA comes. The ribosome will catalyze peptide bond, so you get this elongation, and then goes round and round, adding more and more until you get a full polypeptide which is cleaved off the ribosome. Okay? And then the ribosome can go bind to a new mRNA, and the whole process can start again. So ribosome translates the mRNA. Yeah.
So the ribosomal RNA assembles the protein then?
Very good question. Now that's advanced. That's probably PhD level question. Okay? If you go down here, so if you look at that, there is the RNA, right, in the ribosome. So this is a color-coded structure. The ribosome is an amazing machine. It's made up of both RNA and protein. So the RNA is the ribosomal RNA that's shown here in kind of red color and yellow color. So in the small subunit, we have one large RNA molecule in yellow color. Here we have another large RNA in red color. There's also one here in blue color, purple color up there. And all the ribosomal proteins are in cyan color and magenta color. So if you look at where the tRNA, this is the tRNA, you can see one tRNA, another tRNA, and the third tRNA. The tRNAs are actually buried inside this ribosomal RNA. And it's the ribosomal RNA that catalyzes peptide bond formation. Okay? The proteins are all on the outside. They really don't do much. It's the ribosomal RNA. Clear to you? So it's RNA, ribosomal RNA, that does the real function of the ribosome.
So they are the ones that assembles it, basically?
Yeah,
Or-
they are the ones that link the
The
amino acids
amino
together.
acids,
They
yeah.
join the amino acids together. That happens right here in the heart of this machine, which is mainly made up of RNA. So scientists think it's the RNA that does the assembly. Okay, so here is the overview. So you have a long mRNA. It's not that just one ribosome will bind to the mRNA. Many ribosomes can bind and they can do parallel processing. Okay, just like computers with multiple cores, right? These days everybody wants to buy quad core and things like that. Same way, the cell won't waste any time. You have multiple ribosomes. So you have one ribosome, another ribosome, another ribosome, another ribosome. They're busily translating the information in the mRNA. They're making protein. So they assemble amino acids together. So for example, if you look at this ribosome, this is towards the end of the mRNA. So it has attached a lot of the amino acids together to make a long protein. This is just starting. So this just has one amino acid. As it travels along the mRNA, it's going to link more and more amino acids, so it can form the polypeptide. This is the completed polypeptide, and the two ribosomal subunits will separate. Any questions regarding what the ribosome does? Clear? Okay. Sometimes people, if you look at the cell, you can see something called a Christmas tree, because you have this long chains towards one end and short chains towards the other, so it looks like a Christmas tree, right? It's long. Okay. We're going to look at mutations. What is meant by mutation? We already started talking about mutations. Mutation is any change in DNA. So I already mentioned things that can cause change in DNA, either radiations, chemicals, heat, and errors during DNA replication can cause mutation. And these mutations can take place in genes. If they happen in some very important genes, for example, genes that are important for, I don't know, maintaining your sodium content in the cell, and if that gene is mutated, of course it's going to be lethal or can cause deadly diseases. Okay? There are many kinds of mutations. Some mutations are silent because the change may not change the sequence of the polypeptide, because as you know, the codon is redundant in some cases. But in most cases, they can cause problems. Okay, now I'm going to just quickly refresh our memory. These are the key concepts. Can someone tell me what is on that corner? What is that called?
A phosphate.
A phosphate. What is this called?
The monosaccharide.
Huh?
Monosaccharide.
Monosaccharide. Okay, what kind of a monosaccharide or a sugar is that? It's a ribose. Okay, it's a ribose. What is that?
That is a deoxy.
No deoxy. It's just a base. What kind of a base is it?
Deoxy.
No. There's nothing. There's no sugar or anything like that.
Okay.
So you can't tell anything. You can say, is it a purine or a pyramid?
It's a pure.
It's a purine. Good. Excellent. Okay. So, how is the DNA organized? Is it parallel or anti-parallel? Okay, it's an anti-parallel. Are the bases inside or outside?
Inside.
Inside the double helix. What is on the outside?
The phosphate.
Phosphate and the sugar. Why is it on the outside?
Because those are hydrophilic.
They are hydrophilic and the bases are hydrophobic. Okay, you guys are good. Okay. Okay, we learned about replication fork, right? This is a picture of a replication fork. Which is the leading strand or the continuously synthesized strand? Is the top one or the bottom one? Is it A?
A.
A is leading. What is B and C?
Lagging.
Lagging strand. And what do we call those fragments? Okazaki
Fragments.
Okay,
Fragments.
good. Excellent. If you know all that, I'm pretty impressed. Okay. Now let's go to advanced level. Do you want to go? This is just quickly. So if you're curious, how are these bases arranged? This is just showing how the thymine and adenine comes together. These are the hydrogen bonds that are formed. And they're perfectly complementary. When you have a hydrogen bond donor, you have a hydrogen bond acceptor. Okay? You don't have to memorize anything. Most people don't even know how to draw these structures, but that's fine. But I thought I'd just show you. And this is just the details during DNA replication. You're forming a new bond. So the substrate, the incoming nucleotide comes as a triphosphate, and then this hydroxyl group will link to this phosphate group, okay? And so this provides the energy for forming this new bond, basically. So that's just showing how the bases come together. But that's okay. Now let's move on to something interesting.
Whoa.
What is this?
Influenza.
Influenza virus, right? And what does influenza virus cause?
The flu.
The flu, right? Lots of people die of flu, especially very small children and older people. Now, influenza virus, of course, is an RNA virus. You can see this is the influenza virus. It's got a membrane and it's got the RNA inside it. Okay, it's an RNA virus and it can infect us. People are concerned about influenza because there are some deadly strains of them, okay? There is no vaccine against some of these strains and they can actually be very dangerous. They can kill you. And I think in 2009, there was a pandemic of, what was that called? Bird flu, right? There was a bird flu, then there was swine flu, and so now there are again, I think bird flu is on the rise. And here they're killing all these chickens because apparently some of them are infected with bird flu. So why am I talking about that? Because this is a very small virus. All it has is really 8 small RNA segments. And this small virus with 8 small RNA segments can be so deadly. Okay, all it has is some RNA and a membrane, and it can get into our body and it can cause damage. What is this one? This is HIV virus. This is another RNA virus. You might have heard about HIV, and this is actually, I think, a lymphocytes. Yeah, lymphocytes with HIV budding off this cell. So this is our cell, our lymphocyte, and you can see how the virus packages itself. This is the viral RNA. It buds off, forming infectious virions or infectious virus particles that can be, that we can spread from person to person.
What is the larger body in this picture?
So this is a cell. This is a leukocyte. This is one of our white blood cells. Okay, our blood cell, and they stain it in blue color. And the viral RNA is shown here in yellow color, budding off from the surface. These are free viruses that are emerging from an infected cell. Okay. So this is just the life cycle of HIV. As I told you, HIV is an RNA virus. It cannot survive by itself on the outside. It needs a host. It needs to infect somebody. And it infects specifically this immune cells, T lymphocytes. And once it gets inside the cell, it makes the RNA or the virus is released into the cytoplasm. It's copied into DNA. And it's the DNA that travels into the nucleus where we have our DNA, and it sort of glues itself into the host DNA, into our DNA. And so it's very hard to treat HIV because it has not become part of our DNA. There's no way to find out which is HIV DNA and which is host DNA. And that's why this is a serious disease or a deadly infection. And once it becomes part of our DNA, it starts just transcribing, making more and more copies of RNA, which can then package itself and produce viruses that can infect neighboring cells. It's very devastating. It can kill all the immune cells. That's why it's called human immunodeficiency virus. It causes our immune system to fail, and we can then die from bacterial infection or any other infection.
How do you get HIV?
How do you get HIV? Mainly by blood transfusion. So if you have contaminated blood, if suppose a patient or a person has HIV, and in the 1980s, there was no screen for testing whether that is HIV positive blood. And so many people got infected because of blood transfusion. You can also get by taking drugs with improper needles, needles that are not sterilized. Some of this was transmitted during, by drug addicts. They share needles, they poke themselves, and they give the needle to their friends and say, hey, here, take a shot. And what happens is the needles are contaminated with blood, which may contain HIV. And once HIV gets into your system, it can, of course, bind to blood cells and get inside. So that's why it's very important to have very clean habits and, sterilize anything in terms of surgical procedures. Now, all that is taken care of nowadays. Hospitals make sure that they don't use non-sterile equipments and instruments. So how do you treat a virus like this, which gets into your DNA and stays there forever? I mean, there are some drugs available. One of the drugs that are used are these nucleotide analogs, okay? We talked about nucleotides, we talked about adenine, guanine, thymine, and cytosine. So they come up with drugs that look almost like the real DNA. So in this case, this is like a thymine, right? It's got a metal group, this is thymine, it's got a sugar. But here, if you notice, it's got N3, it's got a nitrogen instead of a 3' hydroxyl group. So why is that a drug for treating AIDS? The reason is that this one cannot be extended by the DNA polymerase enzyme. This is like a dummy substrate. It will get into the DNA and then no more DNA replication. So the virus cannot replicate anymore. And it's making more copies of the RNA. This drug will get into our body and will prevent the replication of the virus. Okay, that's why AZT, azithromycin, is a drug to treat HIV. There's other versions of it. So there's 3TC, which has a thio group here. And again, that can be accommodated by the enzyme, but will not be extended by the polymerase. Yeah.
Are there any like negative effects of these drugs?
Yeah, these, of course, our body also has to replicate the DNA because cells are constantly dying in our body, and so it can cause a lot of side effects. So the dosage has to be adjusted very precisely, otherwise these people go through a lot of trauma. So that's why these are not good drugs, but hey, what can you do? The options are, if you have AIDS, you're going to die soon, but if you take these drugs, it can prolong your life 10 or 20 years. There are new classes of drugs now, they're called protease inhibitors, and they also inhibit specific enzymes that the HIV virus use. So I think that's all I had in terms of my lecture. So if you have any questions, I'll be more than happy to chat with you. Okay. So, what have you learned so far? Any questions?
Wasn't it, what was her name? Rosalind Franklin?
Rosalind Franklin, yeah.
Wasn't she the one who actually discovered the double helix instead of?
She did the structure. She
And then they copied her.
That's a big controversy. She didn't get the Nobel Prize because she died before the Nobel Prize was given. And one of the rules of the Nobel Committee is that the scientist has to be alive. Rosalind Franklin was the first one to have the diffraction pattern, they call it, of this DNA crystal. Okay?
Didn't she die because of the radiation, because of her work too?
It's possible that she was exposed to a lot of radiation because she's working with the X-ray machines. And so she was the first one who got this beautiful DNA structural pattern. And illegally, or I don't know how, Maurice Wilkins, who was Rosalind's boss, gave information to Watson and Crick. And it was a mad race to solve the structure of DNA. Actually, I had a movie, I can show you that, but I won't project maybe.
And then didn't they do it, but she like fixed it and?
No. These guys, what they did was, by looking at the image, it was very clear to them what the structure is because they could see the helical pattern. And they arrived at the structure before Rosalind Franklin did. And these guys didn't actually do any experiment. They used other people's experimental data to arrive at the structure purely in a theoretical fashion. Okay? So that was interesting story there. And there's a lot written about them. You can check it out in Wikipedia. Many people feel that she should have been given the Nobel Prize along with them, and I agree with that. She was proud. She had done remarkably well in terms of solving many structures. Let's see, if I can show you this movie, that will be nice. We don't have much time. It's just a few minutes just to excite you about DNA. Let's see whether it works.
Takes them.
He'll see immediately.
I actually have to go to the next class. Is that okay?
50 years ago, two scientists made a discovery that would change the world.
Okay.
Jim Watson and Francis Crick revealed the structure of DNA, the single molecule that carries the genetic instructions to build all living things.
I don't know why it doesn't show.
We've done something really important. We have discovered the secret of life.
But how